There are three main parts to cellular respiration: glycolysis, the Krebs cycle, and oxidative phosphorylation. Glycolysis occurs in the cytoplasm, breaking down glucose into 2-pyruvate. This process uses 2 ATP at the beginning but produces 4 ATP in the process, with a net result of plus 2 ATP. The 2-pyruvate are converted into 2-acetyl in the mitochondria, and they each bind to one coenzyme A to form 2 acetyl-CoAs. The coenzymes escort the acetyl groups to the matrix of the mitochondria, initiating the Krebs cycle. I'm not going to cover the specifics of the Krebs cycle here because it gets quite complicated and there are a lot of steps. But at the end of the Krebs cycle, the net result is 2 CO2, 3 NADH, and 1 FADH2. The last two molecules here are not the same as NADPH or NADP+. Those are for photosynthesis. We get these products for every acetyl that goes through the cycle. So since we started with one glucose that's split into 2-pyruvate, we end up with 2 sets of the products here. Now for oxidative phosphorylation. In the inner mitochondrial membrane, NADH and FADH2 are oxidized, releasing their electrons and energy. This turns the NADH and FADH2 into NAD+ and FADH respectively. The NAD+ and FADH are recycled back to be used in the cycle again. The released electrons move through an electron transport chain, eventually combining with O2 and protons, which are the H+ ions, to form water. The electrons release energy as they move through the ATC, which gets used to push protons through the enzyme ATP synthase to produce ATP. This is the same kind of system that was in photosynthesis, called chemiosmosis. If you're curious about that, you can watch my video on photosynthesis. Chemiosmosis in cellular respiration generates approximately 32 to 34 ATP. When the cells do not have access to O2, they cannot go through the normal process. Instead, some cells will use a different process called fermentation. There are many types of fermentation, but the two most common are alcohol fermentation and lactic acid fermentation. Note that glycolysis still occurs here since it is independent of O2. Alcohol fermentation converts pyruvate into ethanol, releasing CO2 in the process. In lactic acid fermentation, pyruvate is converted into lactate, which oxidizes NADH in the process. That means the NADH loses one of its electrons to form the lactate. That's all I wanted to cover in this video. Thank you for listening to my explanation. If you like the way I explain things, do subscribe to the channel, as I will be posting more videos like this in the future. If you're confused about anything, feel free to leave a question in the comments. I'll be sure to answer. And if you're curious about something else, I can answer questions about that as well.